Morning, everyone. Thanks for signing up the training either on-site or by Zoom. I'm Tianyu Li. I'm the statistician working at the breast cancer group. Today, Nabia and I will talk about this Bayesian optimal interval design, its implementation and application in the clinical trials. So for the first 1 hour, I will cover these topics. I will give a brief introduction of the bone design and how to generate it, how to draft it in the protocol, how to monitor the design, and eventually how to get to the MTD. And then I will also talk about other applications of the design in the clinical trial. Then Nabia will take over to focus on this blue panel. She will talk more in details about the statistician's role in monitoring toxicities in clinical trials. So first, the bone design. The bone design basically is a phase 1 design. A phase 1 design of cancer treatment trials, usually we evaluate the toxicity of a drug by observing the DLT. The DLT is the dose-limiting toxicity, which we need read it up in the protocol in advance. And also we do dose escalation or de-escalation across different level of doses. Then eventually, if we have luck, we will find the MTD. That is the maximum tolerated dose. Using this MTD and combined with other data information, we will finally decide on a dose that we will use for the phase 2 or phase 3 design trials. So a good phase 1 design method is the one that can enable us to pick the true MTD. So in the past 3 decades, starting from 1990, 3+3 design, which is, I think most of people heard about that. The 3+ design have been, okay, should be this one. The 3+ design has dominated the phase 1 clinical trial due to a couple of very desirable features. But first, I will just go over what a 3+ design is. So first, we enter 3 patients to a dose level, and then if we observe no DLT, then we escalate to dose level +1. If we observe 2 or more DLTs among these 3, then this dose level is too toxic. The dose level -1 will be the MTD. If we observe 1 out of 3 DLT, then we add 3 more patients. So in total, we will have 6 patients in this dose level. So among these 6, if we have 1 or less DLT, then we escalate to dose +1. If we observe 2 or more DLTs among the 6 patients, then the dose -1 is MTD. Then the design will be stopped. That's it. So we can see from this flow chart, it's very easy, very simple. So that's why the clinicians really love it because it's simple. You do not need any stat software. You can just follow it. So very friendly to clinicians. And also it's transparent because all the rules is in this flow chart, nothing else. So for the regulatory institutions like RRB and FDA, when they see a 3+3 design, they are so familiar with it, so they can do a quick approval. But still, there are some disadvantages of this design. First is inaccurate MTD estimation. It usually will identify the MTD that's lower than the true LTD. So that means we will move an underdosed drug to phase 2. The patient won't get enough treatment. And second, we can see here, we cannot preset our DLT rate. It's what's there is there. So it's not flexible. So in the recent years, the bone design starts become more and more popular because the bone design have a couple of really good features. First, it has the higher accuracy to identify the MTD. For this point, we will talk more when I illustrate how to do the design. So since we can accurate to identify the MTD. That means we can minimize number of patients treated at underdose or overly toxic drugs. That make this design is ethical and efficient. Also, for this design, we can preset a DLT rate based on our previous medical experience or literature research. So it's flexible. And also, it's in reality, it's actually very easy to follow. We will have a preset boundary table, which I will also show you when I illustrate how to do this design. Still, some clinicians are reluctant to use the bond design because first, they think it requires statisticians' involvement. But this concept actually is wrong and very harmful. The clinical trial, although it is called clinical trial, but actually it's not just simply clinical or medical. It requires a significant amount of involvement from statistician. And second, they do not like it because it requires continuous monitoring. And also the word like Bayesian just make them sound very intimidating. But even though it sounds like Bayesian and model-based, actually it is not that complicated. I can show you later. So actually, if we look at the cons, the three cons, they are actually not really disadvantaged. That's exactly the factors that can make a trial a good trial. That statisticians' involvement and continuous monitoring. So let's see how to generate a bond design. There are a couple of different ways to do that. I know there are some R code you can write and generate a bond design. But in our group, we usually use this website. Also because it has a couple of very desirable features I can show you later. So for this website is created and maintained by MD Anderson, also by the author of this bond design. So for this website, it also gives us the option to do either a single agent design or combination design. So the single agent that we only care about study one drug, but sometimes the PI, they want to have the two drugs and want to see what dose level of this combination will be the MTD. So I will show you, first I show you this website. Here is the trialdesign.org. It needs registration, but you just simply give the email. No password is needed. So the phase one, we can click. Here is the bone suite. That's the bone design we need. It launched here. Launch here. So that's the layout of the website. There are different tabs which they can enable us to do different steps of the design. I can show you with the illustration one by one. So here I have an example. First, we will talk about a single agent design. Just imagine we have a new drug. We have 5 dose level we want to explore. And we want to start at dose level 1. But here, you don't have to always start from dose level 1. You can start from dose level 2 or level 3. That decision is trial specific. It's really up to the PI whether they want to treat the patient at starting with a conservative drug dose or less conservative. So it doesn't matter which level you want to start. And for this drug, my target DLT rate I set as 30%, and I have enough money to fund 45 patients in total. So we can go back to this website. The first type, trial setting, is where you enter all your parameters. And for the trial name is optional, but usually I enter one because for the following tabs, they want to just trace back to this one. So it'd be easier if you enter a name. So for today, since my favorite event will have a grand opening tomorrow, so I will name it World Cup. So, and then you have the option either to type in your parameter or use the previously saved input. Since it is a new design, then I will type in. Just like I mentioned previously, for this drug, we have 5 doses level, and we start from dose level 1. So here you can see you can not necessarily to start from dose level 1. You do have choices here. And the cohort size is how many patients each time you want to enroll to a single, to a certain drug level, to a dose level. The default is 3. That's also what we usually do. We want to observe 3 patients at each time. So here is 3. Since our total is 45, so the number of cohorts will be 15. 3 times 15 is 45. And this bar is where it allows us to choose the cap of the number of patients you want to treat at a single dose level. Because sometimes you don't want to stay at one single dose level forever, like to all the 45 patients. So you want to set a cap here. And also this cap, where you set it, is also the trial specific. For example, for most of the for the phase 1 trial, the goal is to assess toxicity. But still for some trial, they also want to evaluate efficacy. So if you want to evaluate efficacy, you want a larger sample size for that dose level. So you want to set this cap to be larger. But for my study here, I only want to evaluate toxicity. So I pick at 9. This is where you put your preset DLT rate. Is 0.3, is 30%. We don't need to change here. The bond design also give us an overdose control. That is when the software, you tell the software it is too toxic, then we need to stop this dose level or stop this trial. Then how to decide it? It is the posterior probability that given the current data that the probability of DLT is greater than 30% in my case, but it is 30% is what your preset DLT rate. So when this posterior probability is too high, the software knows, okay, we cannot proceed, we should stop. So the default, the probability is 95%. But the CTAP used to just ask to be as low as 75%. But now they changed their mind and they ask to increase to 85%. But here, I still use the conservative one. So I will set at 75%. Then after we enter all our parameters, we click Get Decision Table. Then here is the output. There be also 2 tabs. The first tab gives us a design flow chart. This actually shows us the algorithm of this design and how the design will be carried out in a more mathematical way. So we start as the pre-specified starting dose. We treat 3 cohort of 3 patients. Then a first check if the stopping rule met, that is the dose control, the overdose control. If it's met, it's too toxic, then we stop the trial and select the MTD dose. If it doesn't met, then we compute the DLT rate at the current dose and compare that DLT rate with 2 boundaries. So for this DLT rate is calculated as total number of patients who experience the DLT at the current dose divided by total number of evaluable patients treated as current dose. So here, please remember evaluable. We only take care of evaluable patients. And what is evaluable patients is also should be written in your protocol in advance. Usually for us, a patient have to receive at least 80% of the treatment, the dose, in order to be evaluable. But there can be other criteria, but still, whatever you have, write it in the protocol in advance. And then now we have this DLT rate. We compare with two boundaries. So the two boundaries, they're the two estimates that minimize the average probability of making a wrong dose. That is the misclassification error. So if our DLT rate lower than this lower bound, that means we are safe. This dose level is safe. So we want to escalate the dose to a higher level. If we are higher than the upper bound, then that is bad. Then this tox is too toxic. We want to de-escalate the dose. If our DLT rate is between this boundary, that means we want to evaluate this dose level further. So we will enroll another 3 patients, a new cohort. Then we will repeat the whole process again. For now, it still seems very intimidating because it requires a lot of calculation. We need to calculate DLT. We need to calculate these two very unfriendly boundaries. But however, in reality, it's not that hard. We don't have to do that by ourselves. The bone design already gave us a very easy follow table here. So for how to interpret this table, the first row is the number of evaluable patients treated. That's the evaluable patient at each dose level you monitored, you evaluated. So we can see the highlighted like 3, 6, 9, 12. And then the rest of these little numbers, they are the DLT we observed. For the header of the row is that if you observe a DLT here among the number of treatment, what's your decision? So for example, if we take the first column as example, we treat 3 evaluable patients, we evaluate them, we observe 0 DLT, then the decision is escalate. If we observe 1 DLT among these 3, our decision is to stay. Stay means we will enroll another cohort to evaluate more. If we observe 2 DLTs among these 3, then we de-escalate to a lower level. And in our case here, the 2 out of 3 also means we need eliminate this dose level at all. We won't escalate to this dose anymore. So we can see there are other columns, other options. I will also give an example to further just illustrate how we use this table. Besides table, if you love plot or figures, they also give it a figure here. It's exactly the same thing. The x-axis is the number of evaluable patients, and y-axis is the DLT, number of DLT. And E, the green E means escalate, the blue S means stay, the pink D means de-escalate, and the DE in red means warning. That is means you need eliminate this dose. Dose elimination it means. So we go back to our slides. Before we go to an example, there are some important notes here. So eliminate, the one eliminate we miss. We eliminate the current. We not only eliminate the current dose, but anything higher will be eliminated. And when we eliminate a dose, we automatically de-escalate the dose to the next lower level. But when the lowest dose is eliminated, stop the trial for safety. In this case, no dose should be selected as the MTD. If the current dose is the lowest dose already, and the rule indicates dose de-escalation, because we know we have nowhere to de-escalate. So we need to treat the new patient cohort at this dose level. If the current dose is already the highest dose, and the rule indicates dose escalation, but we have nowhere to escalate, so we treat the new patient cohort at the highest dose. If none of the action, so escalation, de-escalation, elimination is triggered, that means stay. So we treat the new patient cohort at the current dose level. Then we repeat the whole process that, like the flow chart show us, we repeat the whole process until two conditions is met. Then we stop the trial. What are the two conditions? One condition is maximum sample size is reached. In our case, is 45. Or the number of patients treated at current dose reach the cap. In our case, is 9 patients. And the decision according to the table is to stay at the current dose. So please keep the second condition in mind, because I will talk about it more in my example. So now we have our design ready. We want to ask the very key question. Is it a good design? Remember at the beginning of the talk, a good design for phase 1 is the one can pick the true MTD. So does our design, is that good? So then we go back to this website. It gives us a simulation. If you run the simulation to answer these questions, still we give the name, since we already gave the name, so we don't have to enter all the parameters again. The bone design usually, it already gave us the default of different scenarios. So we can see, if we look carefully, we can see for each scenario, it put our true DLT rate, which is 30%, under one of those level. So they are all here. And then there will be a smaller DLT rate, and there will be also larger DLT rate. That's the default. If you do not like it, you can change it. Just double click and you can change your own number. It's also trial specific. You get all these numbers. You want to test it, compare those numbers. It might be because your medical experience or other literature research. So I will use the default here and run simulation. So here is all our simulations. For each block is the result corresponding to each scenario. We take scenario 1 as an example. So in this block, the true DLT rate, that's all the rate we put in previously is put here. So here the dose 1 have this 30%. It is our true DLT rate. Then it gives us the selection probability. That's the probability which dose are more likely to be selected as the MTD dose. We can see in this design, we have 47% probability to select dose 1, which is our true DLT. The third row gives us the proportion of a patient will be allocated to each of the dose level. We can also see for dose 1, we'll have the largest allocation of patient. That is a good thing, because a good operating characteristics means that we have higher probability to pick the true MTD, and we allocate more patient to that dose level. So we can also see that pattern stay the same in other scenario. For this one, we have larger selection probability and almost equally high proportion of patients allocated is also here, and here. So now we know it's a pretty decent trial. And this simulation also give us the estimated number of patients we are going to use for this design. So we can see for different scenario, the estimate range from 12 patients to about 23. So we don't have to use up all the 45 patients we preset. So now we have our design, and we think our design have very decent operating characteristics. Then the PI just asks you to write it up in the protocol. It sounds very intimidating again, because we can see there are a lot of information. But with this website, it's not that hard. It's actually very easy. So we can see the third tab, the trial protocol. We click here. We can see they have different options for you. You can generate a template in different format, in HTML or in Word, or even in Chinese if you collaborate with a Chinese company. Very friendly. So I click a Word. So it generate one. I open it because I want to show what it looks like. So here is the template of the protocol preparation. And you can change your trial name. You don't have to call it workup. So we can see this template is already in very nice shape. It have some brief introduction of this bone design, and also it summarize all your parameters. It also summarizes the overdose control. You don't have to do it yourself. It also gives step-by-step description of the algorithm of how this trial will be carried out. Like here, it also gives you the boundary table. They even give you the flow chart here. They even give you the simulation result. So you can show to IRB and FDA your design has very good operating characteristics. So what you do, just do slide edit, then insert to the statistical section of your protocol. Then that's it. So very, very user-friendly. Then now we have our design. We have our design. We know it's a good design. We write it up. We submit to IRB and FDA, and we have luck. We get approved. Then we activate the study. Then what we do next? Next, we need to monitor our design. We need to carry out this design. Then how to do that? Nabia will talk more about that. Here, I will just talk usually what I do. For me, for the bone design, I usually keep a track sheet. Here is the example. That is, I made up this one based on our usual experience. So for this spreadsheet of this table, this big one, I keep record for each patient, what's their DLT and the decision we have. I also keep the decision table here for convenience. Now I will go over this table. So that's a good way to illustrate how we, in reality, we carry out a bone design. So first, we enroll 3 patients because we start at dose level 1. So we enroll 1 to 3 patients to dose level 1. And we evaluate whether they are evaluable patient they are. And then we evaluate their DLT. So we can say none of them have DLT. That's 0 out of 3. We go to this boundary table, 0 out of 3. The decision is escalate. Then we go to dose level 2. We enroll the new 1 to 3 patient to dose level 2. Among them, we see 1 DLT. That's 1 out of 3. We go to this boundary table, 1 out of 3. The decision is to stay. The stay means that we need to enroll another 3 patients to this dose level. So we enroll the dose level 2 patient 4, 5, 6, and so there is no DLT. Then combined to the previous 3, that's 6. 1 out of 6 has DLT. So 1 out of 6 from this boundary table, the decision is escalate. Then we go to dose level 3. We enroll the new patient 1, 2, 3 to dose level 3. But we can see here, the number 3 of dose level 3 is not evaluable. In that case, we need to replace the patients. So we enroll the number 4 patient to dose level 3. But evaluable patient in this dose level is still 3. And we can see we have 2 DLTs. So 2 out of 3 from our decision table, 2 out of 3 is de-escalate. So we go back to dose level 2, the brown one. Then we enroll the number 7, 8, 9 patient to dose level 2. Then we observe 1 DLT. So remember for previously, we have 1 DLT already. We combine this one, that's 2 DLT out of 9 in total. We check 2 out of 9. The decision is escalate. But however, we know that for dose level 3, it's 2 out of 3. 2 out of 3 means not only means de-escalate, it also means eliminate. So we cannot go back to dose level 3 anymore. Then what we are supposed to do? Because here, remember, one of the conditions I mentioned previously, we stopped the trial for 2 conditions. The first, we reach the cap of 45, the total number of patients, or we reach the cap of the number of patients we can be treated at a single dose. But for that condition, it has an and condition. The and condition is that, I can show you again. The and condition is that we reach this cap, but the decision according to the table is to stay. Apparently, we do not satisfy that condition because our decision is escalate. So based on this condition, we still need to enroll more patients, even though we reach the cap. However, in reality, people do not do that. Most of the time, people will stop at 9 at the cap number, then stop the trial. Because if you keep enrolling patients, you may be stuck in that condition forever. You will stuck in that condition that you cannot escalate, but your decision is not to stay. So you have to use up all your 45 patients. That's kind of like a waste of resources. So for most of the study, they really stop there. But we open to discussion from this one later in our talk. But for this one, since it's an example, I want to strictly follow their rules. So I enroll another 3 patients. I enroll the number 10, 11, 12 patient to dose level 1, dose level 2. Then I get observe 1 DLT. So combined with all the brown blocks, we can see we have 3 DLTs among 12 patients. And 3 out of 12 from decision table is stay. So now we meet the standard, we meet the condition, we can stop the trial. Then I will make a summary table here. So for dose 1, we treat 3, 0 DLT, 2, 12 patients, 3 DLT. Dose level 3, 3 patients, 2 DLT. Then what we do next? With all this information, now it's time for us to generate MTD. Then how to do that? We go back to the website. There is a tab here, select MTD. So first we need to let them know we are still using the World Cup. Then we enter here, we enter the numbers just as what what we have in the summary table. So for dose level 1, I treat 3 patients and 0 DLT. Then dose level 2, 12. Then we have 3 DLT. Then dose 3 patients, 2 DLT. But still remember, even though we never go to dose level 4 or 5, but we still need enter 0 here. Otherwise, they won't calculate for you. So estimate MTD. Here is the result. They give us a posterior DLT estimate here. That's where we want to look. We want to pick the dose level that this posterior estimate is the closest to your true DLT rate that you preset. In our case, that is 30%. So we can see here the estimate is 25%. That's why the MTD is dose level 2. The other two doses either too low or too high. They also give you a good figure illustration here, the visualization here. The red line is the 30%, your true DLT rate. And the 3 dots are the estimate of your 3 dose levels. So we want to pick the dot that is closest to this red line. But we need to remember that in a lot of situations, in some situations, they actually give us the same DLT estimate. So there will be ties. So when you see ties, what you do? There is also a rule here. So for example, you have multiple dose level, have the same estimate of DLT. If the estimate is lower, is below the red line, that means the estimate is lower, smaller than your true DLT rate. Then you pick the largest, the highest dose level. If the estimate, the multiple estimate are above this red line, that means the estimate is larger than your true DLT rate. Then you pick the lowest dose level. When it's equal, when they are all exactly your true DLT rate, you also pick the lowest dose level. Okay, so that's all for the single DLT. That's from the beginning to the end. Now we have our DLT, usually just based on this DLT and based on we combined with other information, we can get the a decent dose level we can use for the phase 2 design. I will also talk a little bit about the combination dose. So sometimes the PI want to have 2 doses. They want to know which the combination of the 2 dose level will be the MTD. It sounds pretty complicated, but actually it's not. It's what they do is that they decompose this matrix, this 2-dimensional matrix, into a series of like single dose. For each one is a combination of 2 dose levels. So for this one, if we have 4 levels here and 3 levels here, then eventually we'll have 12 dose levels. So they still do the same thing, like de-escalate and escalate among these 12 dose levels. But the major difference is that the direction of de-escalation or escalation, because we know for the single agent, when we escalate or de-escalate, there is only one way. It's a one-way thing. You only have one choice. But for the 2-dimensional, actually you have 2 ways. So for example, if you add this, if you add number 5 here, the combination 5 here, and if you want to, the decision is de-escalate, you actually have 2 directions. You can keep the dose level, the drug A the same, and de-escalate on the dose, on the drug B, you de-escalate to number 4. Or you keep the drug B constant and de-escalate drug A. So you de-escalate to number 2. So it's the same thing for the escalate. You can either escalate to 6 or escalate to 8. Then in that situation, how we make the choice? The bone design also gave us a table called the desirability score. It is defined as the posterior probability that their true DLT rate is located within the stay interval within these two lambda, which in our flowchart previously, we mentioned these two boundaries. And they always want us to pick the dose level with larger desirability scores. Then how to use this table? This table, if we can see that for this column, it's called number of patient, then that number of DLTs, then desired score. So actually, we just look at each cell of that, and we observe in this cell how many patients we evaluated and how many DLTs we observed. Then we consult this table to get the desirability score. So for example, if 3 patients and 1 DLT, the desirable score is 32. So we want to pick the dose level with larger desirability score. If there are ties, we pick randomly or by PI preference. But still, here is just the rule. But it's very trial specific because sometimes, for example, for one of my study, the PI, she really is just interested in one drug. She only want to escalate or de-escalate on one drug. The other drug is just for the backup, or the doses just for backup. If you have nowhere to go, then you have to use the other drug. So in that case, she only want to prioritize the escalation or de-escalation on one drug. But still, if you want to do that, please remember to write it up clearly in your protocol in advance. So always write up your rule very clearly. And so that's all my talk about the implementation of bone design in phase 1 design, in phase 1 toxicity evaluable designs. But besides that, actually we use bone design in other scenarios, in other situations. For example, for the phase 1 expansion cohort, as I mentioned before, the phase 1, we usually evaluate the toxicity of a drug, but sometimes we also want to evaluate the efficacy of the drug. In that case, we usually will enroll more patients. That's what we call an expansion cohort. But so when we do the expansion cohort, while we evaluate the efficacy, we can still use the bone design to evaluate the toxicity of that drug, that dose. And another application our group use bone design is for phase 2 safety lead-in. So what it is, usually for phase 2, we evaluate efficacy. From the phase 1 MTD dose, we get a recommended phase 2 dose. We use that in the phase 2 trial. We evaluate efficacy. But nowadays, more FDA has the tendency to request more and more trials. Besides just evaluate the efficacy at phase 2, they want us to do a safety lead-in. So for the first couple, like either 4 patients, 6 patients, 10 or 12 patients, whatever you think reasonable, FDA want us to still evaluate the toxicity. So that's where we use the bone design. Here is an example. For example, I already have this recommended phase 2 dose, and I want to use that in my phase 2 trial. But we still have the concern that it still might be too toxic. So we will have a lower dose level as a backup. And we want to evaluate the first 6 patients in our study to evaluate their toxicity. And this time we want to evaluate serious adverse event. we think it might be 40%. But still, what is serious adverse event? You need to write it up clearly, define it clearly in your protocol in advance. So we use what the website gave us. We do what I just illustrated. We enter all these parameters. We get a decision table. Since I want to evaluate 6 patients only, so I only care about this column. Here is how we do it. First, for this phase 2 trial, for the first, we will monitor the first 6 patients. These 6 patients will be treated at our regular dose, our recommended phase 2 dose. If 2 or fewer patients have DLT, then that means we think this dose level is safe. So we continue our trial as a regular phase 2 trial. We treat the rest of the patient at this dose level. However, if we have more than 2 DLTs, so just from this table, we can see more than 2 means de-escalate or eliminate. Then we have to de-escalate. So for the next 6 patients we enrolled into study, we will treat them at the lower dose level, our fallback dose level. And among these 6 patients, if we have 2 or less DLTs, then that's good. We will continue our trial at this lower dose level. And these 6 patients will be included in our later efficacy analysis. But the previous 6 won't be in our efficacy analysis because they are not treated at the same dose level, but they will be included in our toxicity analysis. But among these 6 patients, if 3 or more patients have DLTs, then that means even this backup dose, lower dose level is still too toxic. We have to stop the trial for further evaluation. Okay, so that's all my talk for today. Then Nabia will take over after the break.
Perfect.
Okay.
So 5-minute break.
Then after she finished her talk, we will open to questions and discussions. Yeah.
Thanks all. So I'm going to kind of switch focus a little bit from like the technical aspects of how to design the Bowen design and talk more about what is the statistician's role in phase 1 or phase 2 safety lead-ins. So I think a little bit different from maybe your standard phase 2, where there's a little bit more less requirement for a statistician's direct involvement while the trial is ongoing, enrolling, treating patients. I wouldn't say there should be no involvement. I would say our group, for example, that I work in, so the women's cancers, breast and GYN cancers, we actually have a study statistician at every study team meeting, regardless of whether it's phase 1 or phase 2, just to be aware of what's happening in the trial. So the study statistician attends the meeting, is aware where they are, what's going on, any concerns, is able to be there to answer any questions that are related to the statistical aspects of the design, or just that could have an impact on analysis later. So we attend all those meetings, but it is really critical that you attend the meetings. In fact, there was a recent working group put together for Dana-Farber phase 1 to come up with some best practices for Dana-Farber's phase 1, specifically the ISTs. And so this is some of the findings from that working group. They'll be circulated more widely, but I think this is actually one of the first places I think we're talking about it. And so I just got some slides from that working group this morning. But so you'll be hearing more about this kind of thing, I think, and it'll be disseminated to like the clinical groups and things like that. But I thought it's really important to talk about it from a statistical perspective, because the implementation of the dose finding is pretty critical, and it does actually require some back and forth. And although we do our best to write this protocol to be as specific as possible, most of the times things happen in the trial that we didn't anticipate. And so there needs to be some flexibility, there needs to be some adjustments, there needs to be some decisions made. And you don't want them making the decisions without a statistician involved, because some of it has the implications for the design. And so one of the things that the working group is going to be telling people is the best practices is this should be happening across the disease groups, that statisticians should be at study team meetings for early phase ISTs and participate in these study specific meetings. Ideally, they should be happening weekly for these, sorry, I'm just going to stand up, for these dose finding studies. Just because, again, it can be specific. It depends on the population. Let's say you have a really rare population, then you're probably not going to meet weekly because you're not going to have a new patient enrolling every week. But if you have a reasonably large population and pool of patients that you're enrolling from, then you might actually have patients enrolling every week. So it kind of just depends on the study. But they should be regular enough that you are monitoring what is happening to these patients in fairly real time. And the meeting should be attended, obviously, by the PI of the trial, the primary manager. We work very closely with the person who's the project manager, primary manager. They're different names, but essentially the person who is in charge of like the trial itself. They generally run the meetings. The study statisticians, and then if it's a multi-site trial, they should be representatives from all participating sites. And the key thing is DLTs should be identified in real time, which is why, like, even when you have multi-site trials, you do want your participating sites to be present at every meeting, because that is where you find out what is happening to all the patients that are being treated on the trial. So during this meeting is when studies, each patient, they'll go through each patient and they'll say, what are the updates on this patient? What adverse events have been identified? Are any of them DLTs? And then it kicks off this kind of discussion. If the DLTs happen between meetings, then they should be identified. They should be reported via emails. But again, there's usually an additional discussion in the next study team meeting. Okay, here's what the patient experienced. Here, let's go through the DLT definition, confirm that what the patient experienced meets the definition of the DLT. Also confirm evaluability, that the patient met the criteria for evaluability as well. So that is kind of just very critical. And I think, to Tianyu's initial discussion about pros and cons of 3+3 and Bowen, there was this kind of the impression that a 3+3 doesn't need a statistician involved because it's such an easy design. You don't need the statistician there to help you interpret what to do. You just know the rules. I think that is part of the problem. We have received data for a number of 3+3 designs where they didn't follow the rules correctly. And most of the time, actually, that's due to DLTs not being identified in real time. So if you don't have this very intense communication between the study team, the research staff who are collecting information, and the PIs and the statisticians, you may not realize that there was a DLT in the patient until you've already moved on from that patient. So going through this, documenting all the AE logs and the AEs, and then actually saying, did any of these AEs meet the criteria, is very critical to do for every single patient. And so these study team meetings are the way you can actually do that in real time. The reality is that the database that we have for storing our clinical trials data does not get entered in real time. It should eventually get entered and be accurate, but it is not entered in real time enough that we can actually accurately capture when the DLTs happened for these dose decisions. So that happened during the era of 3+3s, and it happens during the era of Bowen's. And so it's just that hasn't changed actually. And by the Bowen design is a little bit more complicated in terms of the table, but it also just makes people aware that actually, implementing this is more complicated. And so we involve the statisticians, and then it becomes more clearer that we should be involved in all parts of this process. So just this is what they've come up with in terms of their recommendations for how the safety event identification and decision workflow should go. And I'll kind of go through it. I was not part of this working group. And so I haven't had a chance to comment on this. I'm actually going to send them my feedback because I have a slight edit to this that I think we should implement. But again, I think it's fairly clear, and we'll kind of go over it. So let me first go over each step, and then I'll come back to what my recommendation actually is for this. Having participated in a number of these phase 1 trials, based on my experience. So the first step is that the research nurse or the treating investigator identifies a potential DLT or adverse event of special interest during the clinical trial patient's visit within this protocol DLT window. And that's the other part that can get a bit tricky sometimes. The DLT windows, what events count towards the DLT window or not. Sorry, I'm just going to move this out of the way. Okay, so this research nurse then communicates the potential DLT in real time via emails, or sometimes it happens that they're reporting it at the study team meeting because it just happened and the study team meeting is that day, so we just talk about it in the study team meeting. But the information has to be communicated in real time to the PI and the staff managing the study, most often the primary study manager. Then the PI determines if the toxicity event is a DLT. And then their current workflow is that if it is a DLT, they connect with the PI, the primary study manager connects the PI and statistician to review the protocol-defined decision rules and to then decide what to do. My recommendation actually is whether or not it is a DLT, if there's an event that could be a DLT, that is when they should reach out to the study statistician. Because we have had discussions where, first of all, the PI and the study statistician should agree whether the patient is DLT valuable or not. Did they receive enough drug? Did they meet the criteria for that? And again, if they don't receive enough drug because of toxicity, that's fine. But if they don't receive enough drug because we've had this example, someone wanted to go on vacation and they had their flights booked to Hawaii in the middle of their DLT evaluation period. And somebody messed up and didn't tell the patient, listen, you can't leave Boston for this period. You have to be here. And so we eventually worked around it and we came up with a plan and everything. But it was a bit of a problem because they were not actually there to complete their DLT window. And it was nothing to do with how they were responding or not, how their toxicities. It was a flight that they had booked to go to Hawaii. And so, these are the kind of things that happen that you never anticipate, but actually have a big impact on this, on the trial. And so connecting with the study statistician to review the DLT language and confirm that it is, and sometimes they're fairly straightforward. The DLT language is clear. The patient meets exactly the criteria of a DLT, and it's an email exchange of, yes, we agree this is a DLT, and so now the next step is this. Sometimes, and what actually happens is there's a gray area of whether or not it's a DLT, and we actually have a back and forth discussion about this. The PI will kind of use you as a sounding board for their decision. And they kind of talk me through this and they'll tell me, okay, this happened, that makes me concerned, it's more severe, but this also happened. And then I'm not clear whether it was actually disease progression that is causing all the problems or the drug. And so I act as almost like a sounding board for the PI as they work through this language. And then if it's still not clear, sometimes we actually go to another physician. So sometimes we'll consult another investigator in the group, and they'll talk it through with them as well and say, here's what happened to this patient. What do we think? Do we think should we call this a DLT or not? Because there's a lot of nuance, I think, in medicine when you're treating patients especially in phase 1 trials where you are treating patients who already have been, often phase 1 trials are run in patients who have gone through multiple lines of therapy, right? Now they're just trying something that's still in early stage. We don't know these are patients that are often have a lot of other comorbidities. So sometimes this DLT evaluation can get really complicated. And so that's why I don't think it's necessary the case that you only, once the PI has decided, yes, this is a DLT, they call the statistician and say, okay, what's the rule? This is a DLT. I think we both need to know, yes, this is a DLT or not. Oh, no, it's not a DLT. And I think that's we should be aware when there are patients that clear the DLT window with no issues. So the reason also for that is the patient can clear the DLT window with no issues. Maybe they didn't have any toxicity, but maybe they also didn't take enough drug for other reasons. And so we have to actually know when it's a true not a DLT, that they've received enough drug to be DLT valuable and they've cleared the DLT window with none of the criteria that would cause us to call them DLTs. So that would be my kind of shift, that actually you don't connect with the statistician only when it's a DLT, you connect the statistician regardless. As soon as, when every patient clears their DLT window, the study statistician is informed these patients, patient number X has cleared their DLT window and is either DLT valuable or not. Is this a DLT? And then that is recorded. The statistician says, yes, I agree, this is DLT valuable. And yes, they cleared the window, or no, they didn't. And then they give, if it's time to make a decision, they can update the study team and confirm, yes, we can make this decision. Or if it's not yet time to make a decision, that's okay. We just keep it on our Excel log tracking, and so we know what's next. And then this is the next part. You follow the decision rules, or if there's no decision rule yet, you continue with enrollment. So we on the BOC group, and I didn't give you the ones that they have in the working group because that's not finalized yet. Like I said, they'll be sending those out and but something along these lines in terms of the study team agenda gets tracked, and then the study statistician is also CC'd on that agenda that goes out to everybody. So everybody is aware, and it's like documented what's going on. And the documentation is really important actually for FDA purposes. So FDA auditing requires you to document all these things much more extensively. So we use this kind of table. Essentially, we'll have the subject ID initials, we'll have the dose level at which they're treated, the date they're registered, the DLT period start date, end date, and then we'll have these columns of were they DLT evaluable, is this a DLT or not, per the PI and then per stats. And so this is our documentation that the PI and the study statistician has connected for every single patient and is in agreement. So usually what happens is we are all at the same meeting, or we do it via email, but there is some discussion and then we come to a consensus agreement. And so this allows us to document that for each patient, we've come to a consensus agreement. And like I said, if there is disagreement, which we've had occasionally, we bring in someone else to kind of like be another voice and kind of discussion. So you keep talking about it basically until you get an agreement. Is kind of like, and you keep working through it. And again, like there's certain things I defer to their medical knowledge. I am not a physician. I don't treat patients, but there's certain things that I kind of like, you have to explain it to me to the point where I can agree with it. And I can defer to your medical knowledge. You have to convince me this is why this is the correct decision. And again, like I usually ask them quite a few questions to the point where then I'm like, okay, I understand. And then we document what is the description of the AE that is meeting the DLT criteria. And again, this is all usually also then in the clinical trials database, but that is not entered in real time. So for the purposes of the actual decision rules, we rely heavily on these documentation. And then we also use our own tracker as well, just because then we have all those other kind of things tracked there that Tianyu was sharing. So I think that's the part that I wanted to kind of emphasize on my end today, was there is a general DFC-wide emphasis on better communication, more structured communication, more continuous communication with study statisticians and the clinical team for phase 1 and any kind of safety monitoring for studies. And there is now some like more structured guidelines going on how to do things. And I think we've all been involved in studies where they come to us at the end of the phase 1 and you start going through the data and you realize, oh, wait, something went wrong here. And then you have to figure out how to write it up in the manuscript and explain it. And so we are trying to avoid that now. But by having very close communication, we have we are really like keeping track of everything that's happening in the trial, and the trial is being implemented as it was intended and designed. And that kind of becomes pretty critical. So I'm going to stop sharing, and we are open to questions, discussion. I think one of the things to kind of kick us off can be going back to what Tianya was raised in her section about the 9 patients maximum, and the decision is to stay. I don't know whether anyone has implemented a Bowen design, whether people realized it. Honestly, we just realized it after Jonathan, like there were some questions, and then Jonathan reached out to Ying and he confirmed that that actually is the case. You keep enrolling past the max. So has anyone else actually got to that where that was actually like they kept enrolling past the max, or did they not realize that they were supposed to? I wonder whether anybody else has experience with this and whether that has something that has happened for them.
Can you, Nabia, can you just, since it's been about a half hour, can you revisit?
Yeah.
Too, because my follow-up question is to that. So if we keep enrolling, how much longer do we keep enrolling?
Well, that's, I think, part of it. So, so do you want me to show the slide again?
Sure. Just
okay.
Let's bring us back to a half hour ago.
Yep. I just want to make sure I'm showing the right set of slides. So it's actually better in the, I don't know, Tianya, do you want to go ahead and share on your side while I?
Okay, yeah.
Yeah, because I don't think I have your spreadsheet ready to pick up. I mean, we are going to potentially run into this on our, so the decision, the way this design is meant to work is, if you are at your highest dose level that you have available to you. So in Tianyu's example, we had eliminated dose level 3 and higher. So highest dose level was 2. And the decision was to stay. But in this case, we had treated 9 patients at dose level 2, and the decision is escalate. So per the Bowen design, we cannot stop yet. We have to enroll more patients and until the decision is to stay, or we hit our max sample size. So I think actually the language of the protocol needs a minor edit, because I think if you want to implement it that way, it should say the number of patients treated at the current dose is at least 9, and the decision is to stay. Because that's, I think, what they intend. And that's how the simulations are constructed. I would say, though, that we have a potential NCI CTEP trial that is going to be in this situation. If the last patient we're going to enroll has a DLT, then we're going to eliminate this dose. We're going to go back down, and we've treated 9 patients, but the decision at 9 patients is to stay. So it means we would keep treating more patients. So we had a discussion via email with the NCI statisticians, and they actually don't want us to enroll beyond 9. They want us to stop at 9. And that is a bit vague in the protocol as written, but so we should be cleaning that up. But I think there is a problem. Like, Tianya, can you open up your spreadsheet? The tracking spreadsheet? So for example, in this example here, if there in the third cohort, 10, 11, 12, if there was no DLT, then you would still be 2 out of 12 and the decision would be escalate. And so you could be in this loop where you just keep enrolling patients until you hit 45, which is actually not a good design. Like you don't want to do that. So I think one of the things we've discussed is that we change the design a bit and we say, we add another cap. We say you keep enrolling. Maybe you go past 9 if they want to, but you don't go that far past 9. Maybe you say you can enroll an additional 2 cohorts, and if the decision is still to escalate and there's no additional dose levels being added, then you stop. Like you don't treat beyond an X number of patients, because that does seem like a waste of resources.
Yeah, and also in this example, and also our real practice, the trial in the real life is that the dose level already treated at 9, but the decision is not to stay. It's already the top level. So we have nowhere to escalate anymore. So it's very possible for us to, and also for this level, it already showed it performed very well. It's less likely, it's very unlikely for us to de-escalate. The only option is escalate, but we do not have anywhere to escalate. Then we will be in this level forever.
So the other question that came up is, and the other reason why you wouldn't want to follow the design as, is often we have expansion cohorts. So the Bowen is designed to incorporate an expansion cohort within the trial itself. So that is kind of like you could have a built-in expansion in this, and like say, like I want 15 patients at a dose level, and then you have your enough patients to do a preliminary assessment of efficacy. Often though, we don't do it that way. And the reason is because sometimes the patients that you enroll for dose finding are a different population than you want to do for preliminary assessment of efficacy. So for example, in the GYN studies, we will sometimes in dose finding, we'll enroll endometrial, we enroll ovarian, we'll like have a mix of basically patients, because for the purposes of safety, and they can receive the same treatment. So for the purpose of safety, it doesn't matter, like, which particular type of gynecologic cancer they have, like within obviously a realm of group, but there's sometimes a little bit of flexibility there. But when you're looking at efficacy, it does matter, actually. It is very important what type of cancer they have. And so you don't want to just have them all grouped together. So the expansion cohorts might be an endometrial cohort, an ovarian cohort for the expansion. And so then, yes, you could use some of your patients, but you'd have to basically like enroll, and you'd have to enroll knowing ovarian cancer patients are being enrolled here, endometrial, and we want a certain number of endometrial, certain number of ovarian. So that's one reason. Another that comes up is that we often will have a lot of additional research being done in the expansion cohort. So in the dose finding, you might not have the same requirements because they already got a lot of patient visits. We might not make them undergo research biopsies, for example, which are invasive. And let's say in this example, you do not need research biopsies at dose level 1. It's probably not a dose level that's of any kind of interest or being studied further. But biopsies at dose level 2 will be very informative. So you don't want to make all the patients in the dose finding undergo biopsies. And sometimes we want sequential biopsies because there's like important things that you find from these sequential biopsies. But that's why you'd have a separate expansion. So once you know what dose level you're at and where you think is the right dose level to be at, then you do an expansion cohort, and then you ask the patients, okay, now will you undergo these series of biopsies that'll help us understand how is this drug working, how is this drug efficacious, who should we be giving this drug to, who is more likely to respond to this treatment. So that would be the other reason why you don't want to use up all 45 patients to implement the Bowen as strictly defined. You probably need a separate expansion cohort actually that you want to further study.
Can someone expand a little bit on like how backfill is used in Bowen designs?
So there is the Bowen backfill itself, which to be honest, I haven't explored yet. It's on my to-do list for this week actually, because I'm going to try and see if I can figure that out for a specific trial that we're working on, a design now, where I want to see whether we want to formally backfill with the Bowen. I would say that the backfill itself is extremely common nowadays, which is it's not actually, I think it's actually very understandable. Our drugs are not the same as they were in like 20 years ago, 30 years ago. This kind of monotone, more drug means more efficacious is not true actually. And so often we are treating patients, when we look for the MTD, we don't need to go that high. There could be efficacy at significantly lower doses. And so the idea of backfill is you enroll, you find your MTD because maybe that's probably close to where you want to be, but you also backfill lower dose levels so that you can get an assessment of efficacy at those dose levels. The practicality of how you do that, I don't think it's very well specified in the protocols we've looked at. We ended up, for one of our trials, where we had a rare population, we actually ended up doing, it was very complicated, so it was a bit of a thing, but like it only had 2 dose levels. So it was very simple. But they actually, it was very simple. There were only 2 dose levels. We always intended that we were going to go back. If we escalated up to the 2nd dose level, we were going to go back and also fill the dose level 1, because there was no reason to think that you would not have efficacy at the lower dose level. So the question is how much efficacy? You can't really answer the question of dose level 1 versus dose level 2 in trials of this size with backfill, but you can get some hints and you can get more information to help you decide and so it's not just efficacy, they use biomarkers, they kind of go very in depth on those, in our case 10 patients, and then say, okay, what do we think? Is there a clear winner here when we look at the totality of all the data we've collected, or should we actually be pursuing both and continuing to pursue both until we get to the point where we can make a formal decision?
Because I was kind of curious on the practical side of things on the timing of when backfill is used, because my initial assumption had been, you would first find the MTD, and then once you realize these lower doses are safe, you can fill in the lower doses. But I read a recent protocol where it seems like you actually fill in the backfill while you're doing the Bowen design. And so I guess I'm kind of confused on how that works.
So I think that is and we're doing it in this trial, I think it's for practicality reasons. So you have patients, there are one of the problems with phase 1 trials, right, is that there are limited slots available. And so patients might come in and there's no slots available because you filled up your 3-patient cohort and now you've stopped the trial. So I think the practical reason for this and what we've done is when a patient approaches you while you're so in our case 2 dose levels, while we're evaluating a cohort of dose level 2, we've already cleared dose level 1, it's safe, we've escalated up. We can enroll the patients that approach us while we're waiting to assess dose level 2 into dose level 1. So why turn them away? It doesn't make sense to turn them away when we know at some point we are going to go back and start enrolling patients at dose level 1. The patient is there, they want to participate in the trial, we'll enroll them now while we're waiting. So it's just like, it's to speed up the trial in some sense.
And so if that happens, so let's say you escalated from dose level 1, you're in dose level 2, you fill in some additional people in dose level 1. How are they used in the final DLT or MTD evaluation? Are they included in the final MTD evaluation, or how does that work?
So I think formally they probably I think you can treat it two ways, and I think the technically, I mean, the trial design software is flexible enough. You could think about implementing it. It's a good question, and I think you can think about implementing it in two ways. You could just ignore them for the Bowen design and the MTD evaluation because they're not part of the dose finding. They're backfill. So for the purposes of the MTD, you can ignore them, and they just go into your almost like a separate thing. However, I do question whether you should ignore them. The way we've proceeded is fairly cautiously. So because I was concerned, we enrolled 3 patients, we cleared the dose level 1, no DLTs. But I told them, do not, let's just keep enrolling in cohorts of 3 when we backfill, because I was concerned maybe we just got really lucky on our first 3 patients, and the next 3 patients we enrolled to dose level 1 all have DLTs, and then we have a problem. That hasn't actually happened, but I was concerned about that. So I was having them proceed slowly with the backfill because I was concerned that what happens if you see a safety signal, because we only treated 3 patients, right? And so, yes, there's a high probability that it's a safe dose level, but we treated 3 patients. There's a lot of variability around that estimate. And so, yes, so when it comes to the end of the trial, I think technically you ignore them. I think for the purposes of the MTD evaluation, I think that I'd have to look at, I think we'd have to look at the implementation of the trial design MTD. So it is the isotonic regression. I think we'd have to look at how that incorporates the study design, and whether that would how much that would make a difference. I think from the purposes from the pure data point, there is an argument to me to say, let's just use all the data you have, right? You enrolled more patients through backfill, why not use their data? It seems a waste to ignore it. But I don't know whether the trialdesign.org website is then a tool you can use. You might have to program your own isotonic regression to do that.
Got it. So just for my understanding, so when you're going through these decision rules, your recommendation would be to just only pay attention against the formal cohorts, but then potentially at the very end, when you finish the Bowen design, you've come to either you kind of reached your final dose, what have you. And in your final table, you might also include all the backfill patients to determine an FTV.
Yeah. Yeah. My only caveat would be if you see a serious safety signal at your backfill, as you're backfilling, if you're simultaneously backfilling, you probably end up taking that seriously and modifying how you do things.
Got it. Yeah, because I wasn't sure, when we're talking about max number of patients per dose level and that sort of thing, I wasn't entirely sure how backfill would affect that. Anyway.
Yeah, I think the way most of them are written, backfill is separate.
Okay.
But again, I guess the question is, should it be completely separate? And so so that was saying, I haven't fully explored how the Bowen backfill design works. And so that's what I'm actually going to. So maybe we could have another session on that.
I have actually seen it once. It could be as Boeing design where they have set up the rule for how to escalate and de-escalate when you notice the conflict between your backfill dose and the actual Boeing dose. But I'm no expert on that, so I don't think I can answer more questions. But it's worth looking into. It's DF Boeing.
Yeah. Yeah, I'm actually going to be looking at it because we have a design where it might be worthwhile to formally incorporate the backfill in the design as opposed to just having it be like a separate add-on.
I do have experience with one of the trial which is I'm working on with Emanuele. So for a lung cancer phase 1 trial, we did the Boeing design, everything. We checked out the MTD. The FDA then requested us to test one of the lower dose level, which was not being tested in the Boeing dose escalation because it was the lowest dose level. And they want to see if there's any difference in the efficacy or not efficacy, but they will be measured in the pharmacokinetics, the biometrics, for example. So we're going to enroll 6 patients as the backfill and compare to the dose expansion, which we're going to perform at the maximum tolerable dose. It's going to be a randomized comparison.
So the reason we are seeing all of this backfill, where the reason why it is such a like almost every phase 1 protocol we review nowadays has it, is because the FDA is pushing it. So the FDA has this Project Optimus, which is this idea, which is, again, it's probably the right approach given that the drugs are so different nowadays, is that you don't just assume when you get to your MTD, that's your dose level and that's the only one you move forward with. They want you to evaluate and be more fully the lower dose levels for efficacy. So because you don't want to maximize your toxicity if there's no efficacy benefit for maximizing the toxicity. And the only way you'll do that is if you enroll more patients at these lower dose levels and evaluate efficacy. And so that's why there is this push, because I think there's a realization across the field that only evaluating the MTD is insufficient, and that the FDA now has formalized this, and so all the designs now incorporate it.
And also, we need to keep in mind the MTD is not necessarily the dose we will use at phase 2, so it's not equal recommended phase 2 dose. That needs more information. So I think in this time, the backfill provides that information. And also, just like Nabia said, FDA kind of like push that. So for many phase 1 clinical trial protocols I have reviewed, now their final candidate is not a single MTD. They even have like a 2 MTD. They have like 2 doses or even more doses. They evaluate more. Then they will decide which one will go to the phase 2. So it becomes not really a linear decision now. It have all kinds of information back and forth.
I think the other thing that has always been the case, and that in terms of that, we have a trial that we're writing up. It's one of the older trials, but patients tolerated the drug reasonably well in the DLT window. The problem is they couldn't stay on it much longer past the DLT window. So you can't just, like, for the purposes of making decisions in a timely manner, you specify your DLT window. But the reality is the toxicities can sometimes be cumulative. And so for those studies, when we looked at the totality of the toxicities and we looked at how long patients were able to tolerate the drug and how often they had to go off treatment because of toxicity, the dose level that cleared safety was not really safe on the long term. And so they did not move forward with that dose level. It was found to be on the longer term not safe.
I have another question for Nabi and Tianyu. So when you said the study team should reach to us when they see a DLT, what if it's not like at the time of the cohort, 3 patients cohort being finalized? So for example, we're looking at this table. If we have 8 patients and there are 3 DLT cases, and the study team come to ask what to do next. Do we tell them to finish this cohort until 9 patients we evaluate, or just follow the table and de-escalate there at 8 patients?
I think it depends. We have sometimes stopped before 3. So for example, I had a trial where in the first cohort of patients at a dose level, the first 3 patients, they enrolled 2 patients, both of them had DLTs. They were like, we're not enrolling the 3rd patient. Like we were eliminating this dose level. It doesn't make sense to treat the patient at this, like expose any more patients. This is too toxic. And they were very happy to, they were confident that they didn't need to treat the 3rd patient because either way the decision was the same. And they could like the actual treating the patients. So I would be a little bit more hesitant once you're later out about that. Sometimes it's for practical reasons, like it's a slow enrolling trial. It's taking really long to find these patients. It could be another 2 months before you find patient 9. And you need to de-escalate. So, well, in this case, you would de-escalate. The decision rule is different for 8 and 9. But sometimes if the decision rule is the same, you kind of like, you're like, okay, I'm already at the decision rule, no matter what happens to this last patient. So there may be a good reason not to do that. The Bowen design, to a certain extent, does offer you that flexibility. You can make a decision at any point. So that is true.
I guess the follow-up question would be if, for example, we de-escalate 8 patients and it comes back to the same dose later on, do we enroll up to 12 patients or 3 as a cohort to 11 patients? Or just evaluate case by case?
Yeah, I mean, I think if you want to do it case by case, then you should have pre-specified that in your design almost. Like you should say then you're going to enroll in cohorts of 1, but that is a little bit of a tricky thing because then you have to wait for each patient. I think you basically if you make a decision early, you should then still only enroll a maximum of 3 patients. I think if you say in your protocol that you're going to enroll in cohorts of 3, you can enroll a maximum of 3 patients before you make a decision. We have a trial now where basically it's safe. We've had no DLTs across any of the dose levels. So we are modifying the protocol to basically get approval that the last group of patients, that in fact the one I was talking about where we only have 2 dose levels. So the last group of patients, we wanted 10 patients in each dose level, and so we had said cohorts of 3, but then we'd have to like enroll 3, wait, and then the last patient would be. So we're modifying the protocol to ask that the last if there's been no DLTs. And we are at we basically have a say escalate. The decision is to escalate. We have then we're that at the final cohort will be a cohort of 4 instead. Because again, I think you have to you have to specify. I don't think you should go above 3 unless you've specified that in the protocol.
Okay, got it. Thank you.
I have a relevant question about the patient replacement. My understanding is that so for Boeing design, there is flexibility. We don't have to replace patient if there is invaluable, especially if many cohorts has been enrolled for a single dose level. So I think for the first 3 patients in cohort 1, might be good idea always do replacement if there is invaluable. But if 4 cohorts has been evaluated, so then I think there is flexibility. I want to get your thoughts on this. In practice, do you always do replacement or consider this flexibility?
I think I agree with you. I think if you've treated a number of patients already at the dose level, and I think if you're planning an expansion cohort that's separate, then maybe you don't replace a patient because you are going to study it further. But I think like in this example we're looking at here, the first 3 patients at the dose level, you wanna replace it because otherwise you only have 2 patients evaluated. And like in this example, let's say for example you didn't replace that 4th patient, you would not have eliminated dose level 3. So your decision on rule would have been very different if you didn't replace that patient. So I think at any dose level, you should treat at least 3 patients. That used to be the part of the, you could not, in fact, I think that is still the case. You cannot make a decision on, they only start the table at 3. You cannot make decisions with less than 3 patients at a dose level.
Thank you.
Okay, thank you both so much. That was great.
Thank you.